Welcome to the Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Please, if you have a moment right now, just pause and like, subscribe, and share this deep dive with friends and family. It really supports the Science Experience. Today, we are deep diving into a paper that, well, it fundamentally rewrites a key chapter in cellular biology.
That really does.
So forget what you might have learned about standard metabolic signaling, during starvation. We're revealing a completely new regulatory pathway.
And at the center of it is this simple fuel molecule, but it's not acting as energy.
No, it's acting more like a molecular handbrake on a crucial cleanup crew inside our cells.
That simple fuel molecule is cytosolic Acetyl-CoA A, or AcCoA for short. And this study proves that AcCoA is a selective signaling metabolite. It controls a really complex process called mitophagy, all through a single receptor.
So our mission today is to unpack this whole intricate system for you. We want to show you the direct, fast-acting link between your nutritional status like fasting and how your cells clean house.
And then how this discovery might just revolutionize the way we approach resistance to certain cancer drugs.
Okay, so let's start at the beginning. You mentioned mitophagy. What exactly is that?
Right. So mitophagy is basically the cell's essential recycling program. It's the selective degradation of damaged or just dysfunctional mitochondria.
The cell's power plant.
The cell's power plants, exactly. It's all about quality control. And it can happen through the well-known canonical PMK1 Parkin pathway or these other more specialized receptor-mediated pathways.
And this paper focuses on one of those specialized receptors, right? Something called NLRX1. What's so special about it?
Well, NLRX1 is unique. It's the only known NOD-like receptor, or NLR, that is located directly on the mitochondria.
So it's right there at the scene of the crime, so to speak.
It is. It functions specifically as a mitophagy receptor, just waiting to bind to another protein called LC3, which starts the whole process of, engulfing and breaking down the damaged goods.
Okay, so that's the cleanup crew. Let's talk about the fuel gauge. Most of us think of AcCoA as being inside the mitochondria for energy.
Right, for the TCA cycle.
Exactly. But this paper is all about the cytosolic pool, the AcCoA outside the power plant. Why is that distinction so important?
It's absolutely crucial because that cytosolic pool of AcCoA is produced differently. It mainly comes from an enzyme called ATP citrate lyase, or ACLY.
Okay.
And ACLY breaks down citrate that's been exported out of the mitochondria. So this production line, it directly links the amount of nutrients available in the whole cell to the concentration of AcCoA right there in the cytosol.
It's like a real-time readout of the cell's nutritional state.
Precisely.
So let's get to the core finding. If AcCoA is the signal, what actually tells this receptor, NLRX1, to start the cleanup?
It's surprisingly simple. The specific trigger is a reduction in the level of cytosolic AcCoA.
Just a drop in the level.
Just a drop. When that concentration falls, the signal is sent and the response is immediate.
The evidence for this seems really solid. How did they actually prove that this drop was the specific trigger?
Well, they induced it in a few different ways. First, they showed that short-term fasting, just overnight starvation in mice, caused this drop in AcCoA, and then, boom, mitophagy.
And second?
Second, they used chemicals to inhibit those key enzymes we talked about, like ACLY. Blocking the production line also triggered the cleanup.
Okay, so if reducing the fuel is the alarm, you should be able to just turn the alarm off by adding more fuel back?
That's exactly what they did, and it's the really elegant proof. They supplied the cells with acetate, which is the cell can really quickly convert into cytosolic AcCoA.
And that stopped it.
It completely counteracted the mitophagy. It restored the normal mitochondrial content. It's definitive proof that this AcCoA concentration is acting like a rheostat, like a dimmer switch.
That's incredible. But what about in a living organism? Does this hold up, say, in different parts of the body during fasting?
It does, and this is where it gets really interesting. With the tissue specificity, they saw this fasting-induced mitophagy in tissues like muscle and brain.
Tissues that need a lot of energy.
Right, tissues that are very sensitive to fluctuating energy. And that response directly correlated with a drop in cytosolic AcCoA in those tissues.
And what about other tissues?
Well, crucially, they looked at the liver. And in the liver, they found no change in AcCoA and no mitophagy response.
Wow. So it's a very local, very specific signal.
Exactly. It's not a global alarm. It's a targeted one.
Okay, here's the part that really made me sit up. When you talk about starvation responses, you immediately think of the big master regulators, right? AMPK and mTORC1.
The usual suspects.
The usual suspects. Are you telling me this massive cleanup pathway just ignores them?
That is the paper's most provocative claim. It seems to be completely non-canonical. They looked for activation of AMPK and nothing. They checked the status of MTORC1 and no effect.
So this is a totally separate alarm system.
It seems to be an entirely separate rapid response system that just bypasses that decades-old view of how starvation triggers autophagy.
That's a revolutionary idea. So if those regulators aren't involved, we have to focus everything on NLRX1 itself. How does it physically sense these changing ACoA levels?
Well, first they confirmed NLRX1 was the key player using a big CRISPR screen, and then mechanistically they showed that NLRX1 has a specific binding pocket, and it directly latches onto Cytosolic AcCoA.
A direct physical interaction.
A direct molecular interaction. And the binding strength they measured, the dissociation constant, was about 6.6 micromolar. Which perfectly matches the reported physiological concentrations of AcCoA in a cell. So this isn't some lab artifact, it's a highly tuned functional sensor.
Okay, so walk me through the on-off switch. How does this binding actually control the cleanup process? Give me the analogy here.
The best analogy is probably a safety pin on a spring-loaded trap. That's the autoinhibition model.
Okay, I'm with you.
When ACoA levels are high GH, the cell is fed. Its happy ACoA binds to NLRX1 and acts like that safety pin. It locks the receptor into a stable autoinhibited state.
So the trap is shut, can't do anything.
The safety pin is in. It prevents the receptor from grabbing onto LC3 and starting mitophagy.
And then when the cell starts to starve, ACoA levels drop.
The safety pin gets pulled, the ACoA dissociates, the inhibition is released, and NLRX1 is immediately activated. It starts forming these larger complexes, and it's now free to bind LC3 and kick off mitophagy.
That is just so elegant.
And they proved it by making a mutant NLRX1 that couldn't bind ACoA. That mutant was just always on, constantly triggering mitophagy, even when the cell was full of nutrients.
Proving that ACoA binding really is the break.
It is the break.
Okay, let's shift gears, because this is where it gets really interesting, connecting this very basic metabolic pathway to a huge problem in medicine: cancer drug resistance.
Right, this is the critical translational part of the paper. We know that KRAS mutations drive something like 30% of human cancers, pancreatic cancer, for instance.
And a lot of research is going into KRAS inhibitors, these new targeted drugs.
A huge amount. But resistance is a major, major problem.
And you're saying the drug that's meant to kill the cancer is actually activating this cleanup pathway.
That's the incredible mechanism they uncovered. They found that these KRAS inhibitors, these drugs, they actively decrease the expression of that enzyme, ACLY.
The one that makes cytosolic AcCoA.
The very one. So the drug lowers ACoA levels in the tumor cells, and that drop triggers this whole NLRX1-dependent mitophagy pathway.
But wait, why would the cancer cell want to clean house? Wouldn't clearing out its own power plants be a bad thing for the tumor?
You'd think so, but it's actually a masterful defense mechanism. When you hit a tumor cell with chemo, its mitochondria get damaged and start producing tons of reactive oxygen species, ROS.
And ROS is bad for the cell. It pushes it towards cell death.
Exactly. So by rapidly triggering this cleanup, the cancer cells get rid of those damaged ROS-producing mitochondria. They lower their own oxidative stress and, well, they survive.
They're using the cleanup system to fuel their own drug resistance.
That's exactly it.
So the therapeutic target then becomes the defense mechanism itself.
Precisely. And the evidence for this is so clear. When they inhibited mitophagy, either by deleting the NLRX1 gene or by using a chemical inhibitor, it dramatically sensitized the cancer cells to the KRAS drugs.
Wow.
The cells that couldn't do mitophagy, when hit with the KRAS inhibitor, showed way higher levels of ROS and much more cell death. It proves that targeting this ACoA NLRX1 axis could actually reverse the tumor's protective strategy.
This study just provides such a beautifully unified model. Cytosolic AcCoA is a selective metabolic signal detected by this one receptor, NLRX1. It links nutrient abundance directly to this huge decision of whether or not to cannibalize your own mitochondria.
It really does. It explains why this pathway kicks in under such different conditions, from, simple fasting all the way to acute drug stress in a tumor.
It's just a stunning display of molecular elegance.
And I think the final provocative thought here for you to consider is this. The ACoA NLRX1 axis gives us a targetable way to reverse KRAS inhibitor resistance, which is huge. But it also raises a much broader question. How many other nutrient-sensing pathways, which we currently think of as just, generating fuel, are actually controlling these highly specialized immune or quality control processes?
Simple metabolites might be the silent masters of our most complex cellular decisions.
They just might be.
Thank you so much for joining us for this deep dive into cellular metabolism and quality control. If you enjoyed unpacking this fascinating paper with us, please be sure to like, subscribe, and share this deep dive with others. This has been another deep dive from the SAN Science Experience.